Okay. Okay, first is two categories. These are called the G protein linked receptors. The reason why they're called that, is this recording? Good, okay. The reason why they're called that is the G proteins, which are trimeric G proteins, directly bind to the receptor, and the receptor stimulates that exchange. The receptor activates these trimeric G proteins. In other words, we can say those receptors act as guanine nucleotide exchange factors for the appropriate G protein. And we're going to look at two of these things here. We'll take a look at some generalities too. First one is the cyclic nucleotide signaling pathway. This is where we're going to activate an enzyme that produces cyclic AMP or cyclic GMP. Those are those nucleotides with a phosphate twist around in a circle. We said before in the class that they're very important because those cyclic nucleotides activate families of protein kinases, the PKAs and PKGs. We'll see that later. The second one of these G protein linked receptor pathways is what's known as a phospholinositide. I'll explain that in a minute. The second one is what we call the phospholinositide signaling pathway. That jawbreaker of a name is referring to a specific type of membrane phospholipid. Not all phospholipids, just a particular type. This signaling pathway activates an enzyme that cuts that phospholipid into two regulatory molecules. One works inside the cell membrane because it's hydrophobic. The other goes in the cytoplasm and does its regulation there. So this pathway is widely used by many different ligands because you got all bases covered. You can influence membrane events and you can influence cytoplasmic events as well. So those are going to be two examples of G protein linked receptor pathways. But we're not done yet. We're going to look at a couple others. What we call the Wrasse Mapk pathway. This one uses a monomeric G protein called Wrasse. This pathway is critically important in development, and it is very important in cancer biology. Most cancers have defects somewhere along in this pathway, and that leads to stimulation of abnormal stimulation of cell proliferation, which is what cancer is. Yes?
What does this say, phospho?
Phospholinositide, I-N-O-S-I-T-I-D-E. And that's the name for a particular membrane phospholipid. Okay? Okay, so that's another pathway we're going to look at. Very important in development and in cancer biology. Another one we're going to look at. Another one we're going to look at is known as the transforming growth factor beta pathway, or TGF-beta for short. This is a very diverse set of pathways used in numerous developmental events. A wide range of our organs, our skeletal structure, our tissues. You can thank their organization to members that use this pathway here. Cool thing about this pathway does not use any G proteins at all. So, look, no G proteins. And we may even look at a fifth pathway if we have the time. But these are the four major ones we're going to look at. Once again, there's about 20 or so different signaling pathways and different cell types, but these are a couple cool examples, good representative examples, and also very important ones. Now, one thing I like to mention, when we describe these signaling pathways, we're going to look at the different components and things like that one by one. But signaling pathways do not act on their own. Many times, if you activate one signaling pathway, there will be some kind of regulatory molecules produced and they can influence other signaling pathways. Maybe you turn one pathway on and you activate a couple others. Or maybe turning one signaling pathway on might inhibit another signaling pathway. So there's a lot of crosstalk between different signaling pathways. Now, just conceptually, we're going to look at these things one by one, but realize they're not separate. They do interact with each other. Sometimes they will activate other pathways, sometimes they will inhibit other pathways. So these are the kinds of things we're going to be looking at. All right. Questions? This is just a quick overview. Now we're going to try to end things partially to get a little set up in the lab and stuff. We're going to maybe end the class a few minutes early and stuff, but we've got enough time to go through a couple aspects of these G protein linked signaling pathways. So let's do this. Okay, in these G protein linked signaling pathways, that's the phosphonolactide pathway, the cyclic nucleotide pathway. In this case, it uses the trimeric G proteins. These proteins bind to the occupied but not the empty receptor. And the occupied receptor directly binds to G protein and stimulates that GDP for GTP exchange. In other words, the receptor acts as a blinding nucleotide exchange factor. The empty receptor will not bind the G protein at all, so G protein sits there staring at you, inactive with GDP bound. But the receptor is occupied, then we can turn this G protein on. Now, the second thing about these pathways, all of these G protein linked receptors are structurally similar. They form a family of proteins. So all of the receptors form a distant family that have key structural features. Aside from the ability to bind these trimeric G proteins, they have one feature that's really diagnostic of them. What happens is this. These proteins, these receptors, of course, span the cell membrane entirely. And what we have here, what anchors in the cell membrane, you have a part in the outside, and then you've got an alpha helix full of hydrophobic amino acids that snakes into the membrane, and then snakes back out again into the cytoplasm, and then you have a second alpha helix going through the membrane, comes back out, a third one, a fourth one, a fifth one, a sixth one, and a seventh one. All of these G protein linked receptors have that feature. What puts them into the membrane is 7 separate alpha helix regions loaded with hydrophobic amino acids. As a matter of fact, when people start sequencing genes and getting the primary structure amino acid sequence of a protein, and they run it through these computer database. If the database looks and says, oh wow, there's these 7 sections of hydrophobic alpha helix regions, what it's going to do is it's going to spit out, this is a probable G protein linked receptor. Now figure out what it binds to and what it activates. So that's a diagnostic feature of all these guys. So we can see that the trimeric G proteins are all related to each other, and the alpha subunit is a distant relative of the monomeric G proteins. And all of the G protein linked receptors are in the same broad family of proteins. The major differences are what's on the outside, that means what kind of ligand can bind to it, and some differences on the inside that determine what G proteins bind to it. But the center of the molecule, the part that goes in and out of the membrane, that's pretty similar from one of these guys to the next. You have a question?
What do you say is
7 hydrophobic alpha helix regions, that's these, anchor the protein into the membrane. Because if you're hydrophobic, you want to be in a hydrophobic environment. And what's better place to be than a nice greasy cell membrane, right? Okay. So that's some basic features of these guys.